Hello friends, welcome to my channel Sam Biology Class. Today we will discuss about the nuclear pore complex. Before that, I request all of my friends, please subscribe my YouTube channel and also watch, like and share my videos with your friends. The nuclear envelope is a double membrane structure that surrounds the nucleus and separates it from the cytoplasm. It consists of an outer and inner nuclear membrane with a perinuclear space in between. The nuclear envelope is also studded with nuclear pore complexes, which are large protein channels that regulate the movement of ions and molecules between the nucleus and the cytoplasm. Nuclear pore complexes, also known as NPCs, are large aqueous channels that penetrate the nuclear envelope, connecting the nuclear interior with the cytoplasm. These complexes are eightfold symmetrical structures. In a typical growing cell, nearly all traffic between nucleus and the cytoplasm passes through nuclear pore complexes. A growing cell has about 3000 nuclear pore complexes that regulate the passage of ions and molecules between nucleus and cytoplasm. This figure shows the structure of a nuclear pore complex. Nuclear pore complexes have a scaffold consisting of three different stacked rings. These rings are cytoplasmic ring, spoke ring and the nuclear ring. Each ring is with eightfold symmetry. Out of these three, the spoke ring is present in the middle. The spoke ring, which is present in the middle, is intimately associated with the pore membrane linking the outer and the inner nuclear membrane. The spoke ring is flanked by cytoplasmic ring on outer side and by the nuclear ring on the inner side. The nuclear ring is anchored to the nuclear lamina. In addition, there is a less prominent luminal ring that surrounds the pore membrane in the nuclear envelope lumen. 8 filaments project outwards from both the cytoplasmic ring and the nuclear ring. While the cytoplasmic filaments have loose ends, the nuclear filaments are joined at their outer ends by a terminal ring. This arrangement of the nuclear filaments form a structure known as the nuclear basket. The cytoplasmic ring is associated with numerous surface particles which are inactive ribosomes. These are the spokes that connect different rings. In addition, the nuclear pore complex also contains a central channel through which transport of ions and molecules takes place from one side to other. The central channel has a diameter of about 40 nanometer and the channel is 50 to 70 nanometer long. Even though the size of the NPCs vary from species to species, its structure is evolutionarily conserved. Nuclear pore complexes can perform about 1000 translocation events per second, shuttling a mass of 100 million daltons. Despite its high molecular mass, nuclear pore complexes contain only 30 different proteins known as nuclear porins. Known as nuclear porins or nukes. Except for three transmembrane proteins that are believed to anchor the NPC to the nuclear envelope, remaining all nuclear porins are soluble. Due to the eightfold symmetrical structure of the pore complex, each of these porins is present in copies of 8 or multiple of 8. Two multiprotein complexes. One is the 10-member Y complex and the other is NUP93 complex. NUP93 complex make up the scaffold of the nuclear pore complex. The cytoplasmic ring and the nuclear ring are assembled from 16 copies of each of the Y complex. On the other hand, NUP93 complex forms the framework of the spoke ring and interacts with 4 nucleoporins whose transmembrane domains bands and fuse the pore membrane. Friends, this is about the structure of nuclear pore complex. Next function. The principal function of NPCs is to facilitate selective membrane transport of molecules across the nuclear envelope. The pore complexes serve as a highly regulated gateway for the transport of molecules between the nucleus and the cytoplasm. This includes the transport of RNA and proteins from nucleus to the cytoplasm and the transport of proteins including DNA polymerase and lamins, carbohydrates, signaling molecules and lipids from cytoplasm to the nucleus. Small molecules are transported through the pore by passive process. On the other hand, large molecules such as RNA and proteins are transported by active process, often involving specific nuclear transport receptors. So by controlling the transport of molecules between the nucleus and the cytoplasm, nuclear pore complexes help coordinating the activities of the cytoplasm and that of the nucleus. Friends, this is all about the structure and function of nuclear pore complex. For more updates, subscribe my YouTube channel, Sam Biology Class.